is Dr.S.Radha, Assistant Professor of Department of Microbiology, SFR College, Sivagasi. Today I am going to explain about the molecular technique that is known as southern blotting. So, the southern blotting technique is considered to be very much prerequisite for many early technique. So, we are going to today discuss about the basic principle, how it works and what are the steps in southern blotting and how we are processing the sample, then what are the scope or application of this technique. So, southern blotting technique is developed by Edward Southern, and this technique is considered to be basically the purpose of identification of a small size of DNA molecule from a group of a molecule or mixture of a solution. So, this is based upon the gel electrophoresis technique, by which the DNA from the gel will be moved to a blotting process, and then that detection process will be done with the help of a radio labeled probe. So, this is the southern blotting apparatus. Here you can see we have the bottom, the tank of that is filled with a transfer buffer, and above that in the platform the gel will be kept. The gel will be covered with a nitrocellulose paper, and below that there will be a nitrocellulose membrane, and after above we will be piling out the crude nitrocellulose paper, a width of 10 centimeter we will be keeping, and this does not require any power supply or electrophoresis. So, we will be keeping above that a weight of 1 kg, and this setup will be left for a overnight process at a room temperature. By this process the DNA in the gel will be transferred to the membrane or nitrocellulose membrane, and this process is known as blotting. After that we will be processing the blotting paper, that we will see next. So, the step that involves the hybridization process, the first step is the restriction digestion of the DNA molecule. So, DNA for which we are going to perform southern blotting, we have to run it in a gel. Before that we have to digest with the restriction enzyme, and we will be getting a series of band. This will be followed by a second step, that is gel electrophoresis, where we will be running the DNA molecule under electric power. So, the third step is known as the denaturation process. The nylon membrane will be subjected to a alkali hydrolysis process, by which the cells will be degraded and the DNA will be exposed, and that DNA will be attached to the blotting membrane. The blotting membrane is either nitrocellulose membrane or nylon membrane, and this will be subjected to a pressure to transfer this by a capillary action. So, the next step is the baking process or blocking process. For this the DNA has to be attached well to the membrane. We will be keeping a 80 degrees Celsius in the oven for a short time, by which the DNA will be well attached to the membrane. After that we will be adding a that is hybridization solution that will has a labeled probe. So, the radioactive labeled probe will be detecting, suppose it has a specificity or complementary sequence in the DNA molecule, that will attach to the DNA, and there will be radioactive, and it will be visualized by autoradiography phenomena. So, the basic application of this process, that is the southern blotting technique, is this technique forms the base to detect all the DNA in the sample, and this forms a prerequisite for DNA fingerprinting analysis, and by which we can solve the paternity, criminal identification and victim identification and all. Other than that, we can locate the gene of interest by cloning technique or recombinant DNA technique, and this is also used to analyze the restriction fragment length polymorphism, by which can we compare the cultivars of two related species. This is also used for identify the mutation and gene rearrangement in the sequence, so that how it vary from the wild strain. And this technique is used in diagnosis of the diseases that cause a genetic defect by comparing with a healthy person with a affected person. So, all the infection agent can be identified very rapidly with this southern blotting technique. Thank you for watching this video.